DNA and Genetics, a comprehensive guide. This document provides an in-depth exploration of key terms, concepts, and applications within the field of DNA and genetics, encompassing fundamental building blocks, molecular mechanisms, genetic variation, analysis, techniques, and diverse applications. By Bharti Kumar. Fundamental DNA terms. DNA, deoxyribonucleic acid, the molecule that carries genetic information in living organisms. It acts as a blueprint for building and maintaining an organism. Gene, a segment of DNA that contains the instructions for building a specific protein or set of proteins. Genes are responsible for a wide range of traits, from eye color to disease susceptibility. Chromosome, a structure made of DNA and proteins that contains genetic information. Humans have 23 pairs of chromosomes, with one set inherited from each parent. Allele, different forms of a gene that can exist at a specific locus on a chromosome. For example, the gene for eye color can have alleles for blue, brown, or green eyes. Nucleotide, the basic building block of DNA, consisting of a sugar, a phosphate group, and a nitrogenous base. There are 4 types of nucleotides, each with a different nitrogenous base: adenine, A, thymine, T, guanine, G, and cytosine, C. DNA structure and function. DNA structure is crucial to its function as the carrier of genetic information. The double helix structure, with its 2 strands winding around each other, allows for precise replication and transcription. The specific pairing of nitrogenous bases, adenine with thymine, and guanine with cytosine, ensures the accurate copying of DNA. During cell division. Double helix, the shape of DNA, consisting of 2 strands that wind around each other. Base pairing, the specific pairing of nitrogenous bases, A with T, and G with C, in the DNA structure. This pairing is essential for replication and transcription. Replication, the process by which DNA makes a copy of itself before cell division. This ensures that each new cell receives a complete set of genetic instructions. Transcription, the process of copying a segment of DNA into RNA. RNA acts as a messenger molecule, carrying genetic information from the DNA to the ribosomes where proteins are synthesized. Translation, the process by which RNA is used to synthesize proteins. This process involves decoding the genetic information in RNA to produce a specific sequence of amino acids, which make up proteins. Genetic variation and mutations. Mutations, changes in the DNA sequence, are a primary source of genetic variation. While some mutations can be harmful, others can be beneficial or have no noticeable effect. Polymorphisms, the presence of multiple alleles in a population, contribute to the diversity of traits among individuals. Mutation, a change in the DNA sequence that can lead to variations in traits. Mutations can arise spontaneously or be induced by environmental factors. Polymorphism, the presence of 2 or more variants, alleles, at a locus in a population. This is a normal phenomenon that contributes to genetic diversity. SNP, single nucleotide polymorphism, a variation at a single position in a DNA sequence among individuals. SNPs are the most common type of genetic variation. Frameshift mutation, a mutation caused by insertions or deletions that shift the reading frame of the genetic code. This can disrupt the production of functional proteins. DNA analysis techniques. Advancements in DNA technology have led to powerful techniques for analyzing DNA, providing insights into genetic makeup, disease diagnosis, and forensic investigations. PCR, polymerase chain reaction, is a widely used technique for amplifying small segments of DNA, while gel electrophoresis separates DNA fragments based on size. DNA sequencing reveals the exact order of nucleotides in a DNA molecule. PCR, polymerase chain reaction, a technique used to amplify small segments of DNA. This allows for the study of specific DNA regions in detail. Gel electrophoresis, a method for separating DNA fragments based on size. This technique is used to identify and analyze DNA fragments in forensic investigations and genetic research. DNA sequencing, the process of determining the exact sequence of nucleotides in a DNA molecule. This provides complete information about the genetic makeup of an organism. CRISPR, a technology used for editing genes by adding, removing, or altering DNA sequences. CRISPR has the potential to revolutionize medicine and agriculture by allowing targeted gene modifications. Forensic and medical applications. DNA analysis has revolutionized forensic science and medicine, providing valuable tools for identification, diagnosis, and treatment. DNA profiling uses unique DNA characteristics for individual identification, while genetic testing analyzes DNA to identify genetic disorders or predispositions. DNA profiling, the process of determining an individual's DNA characteristics for identification purposes. This is widely used in forensic investigations and paternity testing. Genetic testing, tests that analyze DNA to identify genetic disorders or predispositions. These tests can help predict the risk of developing certain diseases and guide treatment strategies. Gene therapy, a technique that modifies genes to treat or prevent disease. This promising approach aims to replace defective genes or introduce therapeutic genes to correct genetic defects. Forensic DNA analysis, the use of DNA evidence in criminal investigations. DNA analysis has significantly improved the accuracy and reliability of forensic investigations. Advanced concepts. The field of DNA and genetics continues to evolve with advanced concepts like epigenetics, genomics, and bioinformatics. Epigenetics investigates changes in gene expression without alterations to the DNA sequence, while genomics explores the complete set of DNA in an organism. Bioinformatics utilizes computational tools to analyze and manage biological data, particularly in genomics. Epigenetics, the study of changes in gene expression that do not involve alterations to the underlying DNA sequence. These changes can be influenced by environmental factors and play a role in disease development. Genomics, the study of the complete set of DNA, the genome, in an organism, including its structure, function, evolution, and mapping. Genomics has led to a greater understanding of the genetic basis of diseases and personalized medicine. Bioinformatics, the application of computer technology to the management and analysis of biological data, particularly in genomics. Bioinformatics tools are essential for analyzing large datasets and extracting meaningful insights from genomic information. Resources for further exploration. For further exploration of DNA and genetics, numerous resources are available, including government agencies, research institutions, and online platforms. The National Human Genome Research Institute provides a comprehensive glossary of genetic terms and educational materials. The FBI CODIS database manages DNA profiles for forensic investigations.